Okay, well, thank you very much. Hello, and thank all of you very much for joining us today for the Zymo research webinar on how we can improve the accuracy and reproducibility of microbiome measurements. My name is Dr. Janina Krumbeck. I'm the Director of Microbiome Applications at Zymo Research. You probably already know what microbiome is. It's the study of microbial communities, the study of how microbial ecosystems that live everywhere around us. And since the first idea of a living cell was described, let me start the laser, here by Robert Hooke in 1665, microbiologists have been discovering this whole world of microbes that surround us in an ever-growing faster pace. From the first notion that microbes can ferment food and produce beer and cheeses, to the understanding that microorganisms can cause diseases by Robert Koch in the late 1800s. Many of the techniques that were developed in this golden age of microbiology in the late 1800s are still being used today for medical diagnostics, such as gram staining, the Petri dish, and the immunotherapy developed by Paul Ehrlich. But the biggest push for the field really came with the discovery of the DNA helix and the discovery of the 16S RNA. Suddenly, we found out that there is this whole kingdom of life that lives with us, because microbes, most of them cannot be grown in the lab, and the sequencing technologies have developed faster and faster, from the first bacterial genome being sequenced in 1995 to the Human Microbiome Project and Ocean Sampling Day. Now we know that there are about 10 to the 12 microbes on Earth, and about 1 million microbes live on and in each one of us. As I mentioned before, 99% of those microbes are not culturable in the lab, yet they have a huge impact on our lives. The tools to study which microbes are present and what they're doing on a large, high-throughput scale are referred to as metagenomics and next-generation sequencing. This term, next-generation sequencing, or short NGS, can be a bit misleading, as NGS was actually developed about 15 years ago, and we are now in the era of third-generation sequencing. And with the development of NGS and the decrease in sequencing cost and sequencing time, we researchers really started studying everything from the oceans to our pets, to ourselves, to agriculture, to the highest point on Earth. You may even speak of a molecular revolution. Using NGS has become the standard method for microbiome analysis, and more than 7,000 new publications per year. We now have a much better understanding of how we as the host interact with the microbes that form a complex ecosystem, how our genes, the environment, and the diet can influence the microbes that live with us and that we share with our pets, and how they may change over time. Researchers have been so excited about the possibilities that NGS has to offer that the microbiomics field has grown nearly threefold by the numbers of citations since 2005. And one of the leading scientists of the field, Dr. Chris Mason from Weill Cornell Medicine, he's one of Zymo's closest collaborators, and he's doing amazing work analyzing the microbiome of cities that we live in, tracing microbes as well as antibiotic resistances. And I will tell you a little bit more about his projects in a little bit. And he also says that it's really indeed an exciting time for the field of metagenomics and the microbiome. However, there's also the limited ability to compare between different research studies, as the different studies use different protocols, which hinders the progress of research because there's the lack of ability to compare between different studies. And let me show you an example of what this means. You might have heard of this story that came out in 2014. A researcher collected a fecal sample and split the same fecal sample into two. She then sent the same sample to two major US companies that offer fecal microbiome analysis and compared the results, which were drastically different. While both profiles agree, that the Bacteroides and Bacteroidetes are the dominant phyla in those fecal samples. The American gut showed that the abundance of Bacteroides is much higher than the Firmicutes, versus the Ubiome profile showed the exact opposites. So just one stool sample, two dramatically different microbiome profiles by two big players in the field. Both companies use very different protocols to process and analyze the data, but the question remains which one of these results is now the correct one. And while this was just a simple single fecal samples, the second example highlights the drastic impact that different protocols have on the microbiome profile on a much larger scale. So here I'm showing the Human Microbiome Project on the left and the MetaHit project on the right, two of the largest projects that have been done to date studying the human microbiome. Just the Human Microbiome Project by itself had a funding of more than 200 million US dollars. So these studies were really a big deal. But just like before, you can see the same trend that we saw in the single fecal sample, where the ratio of Bacteroidetes and Firmicutes is completely flipped between the two studies. And again, they use very different protocols that led to these different results. So why do these problems exist? Why are standards so important? And if you don't have good standards, how can you measure things well? Because just imagine if we would start trying to build houses or machines using no standards, and just imagine not having the correct or same distance between two threads on a nut and a bolt, and you would have serious complications when you're trying to build or engineer anything. This problem was solved 200 to 3,300 years ago until we developed standards. And you wouldn't be able to build any buildings or other constructions without these standards. It would just not be compatible. But for the microbiome field, these standards have really not been established, and they're currently hardly being used at all. And when you take a more detailed look at the microbiomics workflow, it really is not surprising that the results can be drastically different between different protocols. And each step of the workflow can have a huge impact on the final results. I will go over the major causes that cause this bias that we see in the results. And the first major driver of the bias is the sample collection and storage itself. Keep in mind that you are collecting living organisms that are highly diverse. Some organisms might decay or keep growing in your sample. And that's what I'm illustrating here on the right side. They are as diverse as these items I show here, like ice cubes versus rocks versus bread. You would not think that you can ship all of them in the same way. The ice cubes might just simply melt. The rock is probably pretty stable, but the bread, it's going to start molding. Other organisms are going to start growing on it. And just how drastic just the effect of sample storage can be was nicely illustrated in this paper by Costea et al. From 2017, published in Nature Biotechnology, stating that the variation in the microbiome results introduced by storage method was larger than the within protocol variation, which is very drastic. Another paper looked at the similar effect, and they used human fecal microbiome samples and just looked at different ways to preserve the sample by freezing. And as you can see here, the Bacteroidetes drastically decline in the 3 attempts of the protocol. Here's the fresh sample versus a snap frozen one versus one frozen at -80. And organisms like Bacteroides, which lyse easily, already started degrading just by putting the sample into a cold space without any protection. But even if you think, well, I'll just use a protection reagent, for example, like RNA later, which is very commonly used, you should also consider it carefully, because the addition of some of those reagents can drastically change the microbiome by degrading certain easy to lyse members of the microbiome, as shown in this paper. Even major publications face the challenge of sample collection, and that bacterial growth and decay and nucleic acid degradation can compromise the microbiome measurement. This is a case shown by the American Gut Project, and bias was introduced into the sample collection. And after the fact that they had already collected all the samples from all over the world, did the microbiome analysis, they had to edit the data afterwards to reduce a bacterial bloom, specifically of Escherichia coli, which obscured the results from this study. So they found a very creative way of how to deal with this problem, but it would be great to not even have those issues in the first place. And there are ways to avoid them very easily. Remember at the beginning I was telling you about the Metasub project with Chris Mason, whom we closely collaborate with. And for this project, they are collecting the microbiome of different cities all over the world. And they sample those subways and other areas of the city all on the same day. Any bias introduced by storage to the sample while they are traveling from all over the world back to the US would completely obscure the results. And Zymo Research has developed a DNA RNA preservative that makes this kind of collection possible. It's called DNA RNA Shield, and it's facilitating their work by preserving the samples at room temperature at the time of collection, like a snapshot of the microbiome. There is no growth of any organisms or any nucleic acid decay whatsoever, and the samples are stable for up to a month for RNA and for up to a year for DNA without any need of refrigeration, and they can collect the samples in New York, in Qatar, or in Hong Kong all on the same day and don't need to worry about any changes of the microbiome. So it makes it really easy for these researchers to do their work. And Zymo is also the very proud supplier of the Johnson Space Center and their microbiome research being conducted on the ISS. They also use the DNA RNA Shield preservative provided by Zymo Research. So now that we know that there is a way to avoid any issues with sample collection and preservation, we can move on to DNA and RNA extraction. And again, we are dealing with microbes that have very diverse characteristics. Some are really easy to lyse, like E. coli, while others are difficult to lyse, like Listeria. And all of them have very different cell sizes. Some are really tiny, like a marble here, or larger rocks. And just like in this example, you might need different tools to really break open all of these organisms evenly in your sample. Because if you do not break them open and lyse them properly, you will not extract the DNA, and therefore there will be a bias in your sequencing process. And depending on which lysis method you use, the results are very drastically different, and I'm going to show you some examples. So let's say we know the microbial composition of a given sample, like a mock community. We have a mixture of very hard to lyse and very easy to lyse organisms, as represented here by the stone and the eggshells. And here we can see, we know theoretically what we have added, and we're here comparing 4 different extraction protocols. The Zymo protocol, the human microbiome project protocol that I showed you in the beginning with the fecal sample, a power soil kit, and the QIAGEN kit. And the results are drastically different. You see, in these 3 protocols, the easy-to-lyse organisms are highly overrepresented compared to the theoretical composition when we're just looking at easy-to-lyse and tough-to-lyse organisms. When we look even further and see which organisms are there, we can see the overrepresentation of these organisms shown here in red and in yellow, which are easy to lyse organisms like Salmonella and E. coli. And we only know that there is a bias in the extraction process because we know what we theoretically added to it. So this is like a positive control to show the differences between different extraction protocols. The next step of the microbiome project is the microbiome workflow is the library preparation. And there can be bias introduced by the GC content of the microbes that are present, issues with the PCR reaction itself and the development of chimeras, and the primer coverage. Remember, we have, in most cases, a highly diverse microbiome that all kingdoms of life could be represented in. And if the primer does not cover certain organisms or has a bias towards others, they are not represented as they should be because of the nature of the sample. The sequencing platforms themselves also may have some errors or bias towards certain organisms, but I'm not going to go into more detail about that. And then, of course, the last step of the bioinformatic pipeline can have huge impact on your results depending on how the data is aligned, how chimeras are removed, how the clustering is handled, and so on, to name just a few of the steps. And as Dr. Cano put it, relatively minor alterations in the DNA extraction procedure or the bioinformatics analysis can have a very give you a distorted view of what the microbiome might actually look like in your sample. And none of these steps, from the sample collection to all the way to the bioinformatics, none of them is trivial. The workflow is very complex and prone to error. There has been poor data reproducibility reported, which I will go into more detail in a little bit. And there are many current products and many methods that have not been validated. So therefore, the field is flooded with biased data. And the community of scientists is very much aware of this issue. And there has been a call for changing the pipeline and for using positive control, especially since the field has grown so exponentially. But you may think this paper that I'm showing here is from 2015, that's several years ago. Surely things must have gotten better, but unfortunately we're still facing the same issues. This is the paper I mentioned earlier by Costea, published in Nature Biotechnology. And they did an extensive comparison between different DNA extraction protocols. I think about 21 different protocols. And what they found was that the DNA extraction protocol dramatically influenced the conclusion taken from comparative studies, which is very crucial. And they're not the only ones who showed that. There is a flood of papers, and I'm just showing a few of them here, that look at different extraction protocols using the same samples and just comparing what impact the extraction protocol has on the final results. But we as researchers, we really rely on those other publications to conduct and compare our own research. For every reference we use in our own papers, for every grant proposal we write, we do a lit review. And just to put it a little more bluntly, let's just say one study says we found a large gem. Amazing, really great finding. And then the next study says, oh, we found a small gem. But how can we compare between those different studies? How can we compare those results, especially now that we know that every step of the protocol that these different studies may have used can have a huge impact on the results? So how can we draw any conclusions without using a reference, just even knowing what is small or what is large? And that is it's such an issue because measurements are always relative. And once we add a scale to the findings, we can start actually comparing the results. Of course, there are different kinds of scales that we can use. Some might be more appropriate than others. And in the history of our societies, even humankind has not been able to agree just on a single scale, centimeters versus inches. But hopefully the science community can agree at least on one or just two scales of the microbiome measurements, so we can translate the findings between different studies and different results and communicate the findings better with one another. Because keep in mind, every single step of the workflow influences the outcome and may even change the perspective on the matter. And by now, I hope I have convinced you that by using a standard for microbiome studies, we can start comparing different results and that a standard is absolutely needed. Zymo Research has developed multiple standards so far that can help you identify if there is any bias in your own workflow, and can also help compare between different studies if the same standard is applied, all the way from sample collection and preservation to the bioinformatic analysis. This is what one of Zymo microbiomics standards looks like. It's a microbial standard composed of 10 different organisms, 8 bacteria, and 2 yeast. And we have selected those organisms based on a huge diversity. So we have included organisms that are gram-positive and gram-negative, easy to lyse and tough to lyse, some that are particularly large, like Saccharomyces cerevisiae, and some that are very small and can slip through the bead-beating cracks very easily, like Listeria. They have a huge range of GC content, from 15 to 85%. And with this, you can really discover if there is any bias introduced into your microbiome profile because these cells are so diverse. They are offered either as a whole cell to check for every step of the microbiome process, starting from sample collection and DNA extraction, as well as a DNA standard, where it's the mock community mixed DNA, which can help identify any bias in the library prep and bioinformatics workflow. And of course, if you're using this standard as a benchmark to validate your own workflow with, accuracy absolutely matters. Zymo research guarantees that there is a less than 15% deviation from the defined composition that we provide together with the standard, and a less than 0.01 foreign microbial DNA amount. While this was the even abundance standards, we also offer it as a log scale, so you can detect if there is what the limit of detection for your workflow is and what the sensitivity is. Again, just like before, it is offered as a whole cell community and as the DNA already extracted, and the organisms are just the same as I showed you before for the even community. Zymo's newest standard is the spike-in control. So this standard can be used as an internal process control and enables you the absolute quantification of microbes. So usually when you're used to doing 16S research, you provide 10% of my microbes were E. coli, 5% were organism, the other ones. And so by using this spike-in standard, you can translate those relative abundances back to absolute abundances because you know exactly how much you spiked in. It is offered in a high and a low bacterial load depending on what kind of samples you're working with. If you're working with fecal samples, for example, you might be more interested in using a high bacterial load versus urine is prone to have less bacterial communities present, so you would use a low bacterial load for that one. And it offers the internal process control and absolute quantification. So there is no need to add a qPCR step, an extra workload to your microbiome analysis. So Zymo's research is offering a control for the entire microbiomic workflow. Everything from sample collection, DNA, RNA extraction, library preparation, and NGS analysis. So every single step of the workflow is monitored and tested if there is any bias introduced by the workflow. So for the past minutes, I've described the immense challenges faced by the field, but I also showed you how they can be resolved. Zymo Research has built the tools, machines, and equipment to accurately and effectively measure microbiomes. Zymo Research is the only company to have resolved the major workflow challenges faced by the customers and is the only company to have developed a complete microbiome workflow. Zymo has removed all obstacles faced by microbiome research and is offering a solution for every step of the way, from providing standards and solutions for unbiased sample collection, DNA, RNA extraction, library preparation, and we also offer NGS analysis for your data. And by providing a simple, accurate, and reproducible microbiome workflow, we can make an impact on the science being done and on the translation of the science from the lab to our everyday lives. Standardizing the microbiome field and using standards as a positive control is an asset rather than a chore, as Dr. Schrimmel puts it nicely. And Zymo Research is very passionate about data quality and reproducibility of microbiome studies. And in an effort to make well-defined standards and controls available to all researchers, Zymo Biomics has developed the MSci, the Microbiomic Standards and Control Initiative. So with that, free microbial communities, the free standards, are provided to customers all over the world for free to include into their microbiome workflow to see if there's any bias anywhere introduced into your workflow. And we'd be very excited if some of you would sign up for this MSci initiative after this webinar, and we'd be happy to send you those microbial communities. And with that, I would like to thank you very much for your attention. And I'm at the end of my presentation, and I would be happy to answer any questions that you might have.